Thank you very much, Wes. It's a great honor to be a Kirby lecturer. I am not a clinician, or I'm a clinician in training. And at some point, maybe I'll get that wisdom of Dr. Kirby. I am a basic T-cell biologist and interested in mechanisms of vaccines. And over the last several years, I'm going to try to give you a story of how we made some basic scientific observations and then carried them into the clinic, and make some, at the end, just some overall comments of how I view you might, develop vaccines against malaria and TB with some scientific principles. And I should point out to you, I'm not a malaria biologist. Really, in terms of my career, I started this work about 5 years ago, really just looking at the T cells, but I'm still learning. But I think malaria is really interesting. This slide just highlights the global burden of malaria in a heat map from the year 2000 to the year 2015. And so you can see in sub-Saharan Africa that there's been quite a decrease in the amount of malaria over 15 years. And that's primarily due to 3 interventions. One is giving people in their homes insecticide-treated bed nets, so they sleep under bed nets and don't get bitten. 2 is to try to control malaria better by spraying with mosquitoes. And 3 is just treating more people. But despite those interventions, you can see that there's still 200 to 300 million cases of malaria from Plasmodium falciparum and approximately 600,000 deaths. It's the most common cause of death in infants less than 5 years of age from an infectious disease. So despite these enormous public health efforts, we're still left with a lot of deaths. And so I think most of us would agree that the most effective intervention would be a vaccine. And the goal of the vaccine would be to reduce the number of deaths in cases of malaria and severe disease in infants and young children in Africa. 2, there's a secondary gain in terms of those who go to Africa, such as travelers in the military doing work. You'd want to develop effective vaccine. And 3, the Gates has taken on an effort to eradicate or eliminate malaria. And so I think in the context of an elimination campaign, if you could have a vaccine regimen that was highly effective, it would help in the elimination of malaria. So to understand how we would approach this, I have to show the obligatory life cycle slide. And so this is the sporozoite, which contains about 2,000 proteins. And this is what's in the mosquito salivary gland. And so the mosquito bites you into the skin, and those sporozoites, probably on the order of 50 to 100, are deposited either into the skin, or about half of them get into the blood. And they quickly, within minutes to an hour, traverse into the liver. And once they're in the liver, they stay there, and a single sporozoite in a hepatocyte expands 30,000 times. And so over the course of a week in the liver, there's great expansion, and you're completely asymptomatic. 7 to 10 days later, they burst out of the liver and infect red blood cells, and you become clinically symptomatic. So your choke point for mediating prevention of infection would be this stage. Could you prevent the sporozoites from getting into the liver? So you could do that by antibodies. But as you can see, you would need a high amount of antibodies because you need to prevent every last sporozoite from getting into the liver, since a single sporozoite can initiate an infection. And moreover, it would say not only does there need to be a high amount of antibodies, but you could imagine keeping a high amount of antibodies over a long period of time. And I'm going to talk about that's a major drawback with the current vaccine that's likely to be approved. So antibodies could do it, but it's daunting. Once it's in the liver, you have 7 days for a cellular immune response. Most likely CD8 T cells, but it could be CD4 T cells, NK cells, gamma delta cells, to find the parasites in a large organ and kill them all. So you have 7 full days for cellular immunity to occur. And so that's going to be the focus of the talk today. And then once you're into the blood stage, that would be a whole different type of an immune response with different antigens. We don't work on blood stage vaccines. Many other people do, but I think our aim is to prevent them from getting into the blood. So this is called the pre-erythrocytic stage, where it's antibodies and cellular immunity, and there's the blood stage where you get clinical symptoms. So what are the vaccines? So the furthest along is a vaccine called RTSS, which has been developed by Glaxo for over the course of 30 years. It's a single protein. It's the circumsporozoite protein that coats the surface of the sporozoite. And they took that, and there's a group of repeat regions in that protein, and they linked it to a hepatitis B particle. So it's a particle vaccine. So that's terrific. Particle vaccines are, as you know, hepatitis B or HPV are particle vaccines. So they've optimized the immunogenicity of the antigen itself. And then over years, they developed an adjuvant system. And so the adjuvant they've arrived at is something called ASO1B, which is a Toll-4 ligand, monophospholipid A, in a liposome with another immune stimulator called QS21, which is saponin. And when they give those two together, they get very robust antibodies, probably the highest antibodies of any adjuvant, certainly better than alum or any oil you would give in humans. And you get Th1 responses. And when they've gone through phase 3 testing, they get between 30 and 50% of subjects protected up to 1 year. But over 4 years, that protection diminishes. So you have some level. It's the first vaccine that can mediate some protection against clinical symptoms, against a parasite. But I think you would agree that you have a durability issue, and you actually have potentially an efficacy issue in terms of wanting to get perhaps, upwards of 70 to 90% protection. So that's the furthest one along. The European Commission has recommended that vaccine be approved, and the WHO is deciding right now whether to formally recommend it. The second way to get at that's basically mediated antibodies to prevent the parasites from getting into the liver and then possibly having CD4 T cells in the liver, is using prime boost immunization with either a DNA vaccine followed by a viral vector like adenotype 5 or a chimp ad vector followed by MVA. And I would note that these kind of vaccines, this is exactly very similar to what would be used in Ebola, except you'd use a chimp ad3 followed by MVA to give you durable protection against something like Ebola. And here you're aiming at generating CD8 cells. So you want CD8 cells in the liver. And here they used, Adrian Hill has used a small number of antigens, something called ME-TRAP. So one major antigen and a string of epitopes. And when you do experimental challenge, he gets about 20% short-term protection. So you have something based really with one antigen on antibody, something with just a couple of antigens where you have CD8 T cells, and these are the relative levels of protection. And then you go to the entire whole sporozoite. So you take the whole sporozoite, and in 1967, Ruth Nussenzweig, working at NYU, showed that if she took a mosquito and gently irradiated it, the sporozoites were attenuated. They could not mediate an infection, but if she injected those sporozoites into a mouse, they were completely protected. And so they went ahead and they did this in humans. In the early 70s, they did this. This is David Clyde at the University of Maryland. Applied mosquitoes that had been infected and irradiated to people. And he found that if he gave enough mosquito bites, he could get people protected, because you can challenge people with malaria. And the reason why you can challenge people with malaria is you can measure whether they're infected by blood smear, and if they're infected, you treat them, and it's sterilizing. So that was the first proof of principle that radiation attenuation could work with the whole sporozoite. And over the years, over the next 25 years, people like Steve Hoffman and Tom Ritchie showed that in up to, I think they did 30 volunteers over several years, that you could get up to 80% short-term protection with radiation-attenuated sporozoites. A second generation is some very beautiful work by Stefan Kappa, who is currently at SBRI. It's been renamed, I think, Center for Seder, where he takes the whole sporozoite and deletes specific genes where they would get into the liver and then they would arrest. And he has some beautiful data to show that, and he can, depending on which genes you knock out, the sporozoite can go through different stages in the liver, and he can get very high level of protection. So instead of irradiating him, he's leaving him as, he's attenuating him by gene deletion, and they're working on that right now in humans. And then there's a third way to do it, where we actually just infect people with malaria, and you treat them with a drug that kills them at the blood stage. So they go all the way through the liver, and then in the blood stage, you don't get infected because you're killing the parasites at that stage. So you're getting full breadth of immunity in the liver, and that works terrifically well. And that's work by Robert Sauerwein, and if you challenge people, you can get 10 out of 10 protected short-term, and I think he showed 4 out of 6 at 2 years. But the idea is that the whole sporozoite is better than the subunit vaccines. So with that as a proof of principle, I told you that using mosquitoes as your delivery method with irradiated sporozoites, you needed a dose response. You needed up to 1,000 mosquito bites to get protection. And so this served as a proof of principle, but mosquitoes were not a very useful or scalable delivery platform. So, at least right now. So Steve Hoffman, who had spent 25 years running the naval program for malaria and doing conventional subunit vaccines, had decided in 2004 that this was the necessary proof of principle. So he found a company to actually rear mosquitoes under sterile conditions. So it's located across from a hospital in Rockville, and he has a small room. The mosquitoes are sterilely bred. And then he found a technique to isolate the sporozoites from the salivary glands, wash away any salivary material, count them, and then freeze them. So now you had something in a vial that he could quantitate, and it was clean, and the FDA said, this looks fine for a clinical trial. So he had succeeded in doing the dissection in the freezing of the vaccine, so now you had something that was injectable. In this first clinical trial, he had administered in a dose response to 80 volunteers, subcutaneously or intradermally, the vaccine, and showed that it was safe and well tolerated, but the immune responses were quite low, and there was very little protection. So the question became why? The question was, when he did the process of isolating them and freezing them, despite his potency assays, that they were viable and that they could move, he has a series of assays, were basically they're not very potent because he had to freeze them. 2, was the subQ and the ID route not the best route to do it? And 3, did he not give enough? So those were the 3 questions. So really prior to him doing that clinical trial, somehow we met. Actually, we have to figure out how this occurred, but we are reasonably adept at measuring immune responses in complicated situations. And so he said, why don't you take my vaccine and test them in primates, non-human primates? The reason why you use non-human primates is the innate immune responses are more similar to humans. So when you do non-human primate studies, and we do this mostly with adjuvants, it's much more predictive for what would happen in humans. I mean, I think we start with mice and then we go to primates. And so he'd given me the vaccine at 135,000. That was the maximum amount in the dose, and said, see if you can measure immune responses in primates, so the T-cell responses, which no one had really ever characterized sporozoite-specific T-cell responses. So he gave me the vaccine, and we set up a study, and we gave monkeys, a group of monkeys, subcutaneous injection at 4-week intervals, similar to what they had done in the clinical trial. And then, as, quote, a positive control, we gave the vaccine intravenously at 4-week intervals, but in the same schedule. And then in the blood, and ultimately when we euthanized the animals at the end, in the liver, we characterized their sporozoite-specific T-cell response. And so what you can see is this is from the blood. This is after the 4th immunization, 2 weeks after the 4th immunization. We take the peripheral blood out. These are individual monkeys, and we re-stimulate them with the vaccine, the whole sporozoite, and we measure, are they making sporozoite-specific CD8 or CD4 cells by flow cytometry. What you can see is, had we given the vaccine subcutaneously and measured, these are individual animals, interferon, gamma, IL-2, or TNF, responses were quite low for CD8s and CD4s, very similar to what you had seen in the human trial. Had you given it intravenously, you can see that the responses are much better, ranging from 0.1 to 1%. And they make interferon gamma for TNF, and they make all 3 cytokines for CD4. So that immediately told you that route was important, that IV was much better than sub-Q, and that you had robust CD8 and CD4 sporozoite responses. What was far more dramatic is 4 months after the final immunization, the animals were euthanized and we took the livers out. That's the target organ where you need T cells. That's where they get to first. What we found was all of the animals had actually very high T cell responses by the IV route, but again, the sub-Q route, they were very low. And you can see now you have, of the liver, 3% of their CD8 responses were sporozoite specific for CD8s, and they all made interferon gamma. They don't make the other cytokines. And this is what happens as cells migrate to tissue organs. Often they adopt a different qualitative response. Interferon gamma is the cytokine that mediates killing of malaria. And you can see that CD4 cells were also high, and they express also lower levels of IL-2 and TNF. So we had achieved, I think, what we had designed to do is have a high level of sporozoite-specific T cell responses in the liver by giving the vaccine by intravenous immunization. Finally, just on a little bit of a vaccine point, we did a follow-up study, and we had been giving the vaccine monthly. And I think, you start with that. And in the follow-up study, what we did is, rather than give them monthly, between the third and the fourth immunization, we actually, between the fourth and the fifth immunization, we actually waited 8 weeks, thinking that the T cells might rest down, and when you come in with a vaccine, they would expand better. And indeed, in these animals that had got a monthly, when we waited 8 weeks, there was a nice expansion of CD8 cells. And when we gave the sixth immunization a month later, they actually went down. There was really no effect of the spacing for CD4 cells, but this told us that intervals might be important between immunizations. And we actually got these data in the middle of a clinical trial we were doing, and we actually changed the clinical trial to eliminate one of the immunizations and put a spacing between the fourth and the fifth immunization of 8 weeks. So this tells you the flexibility we have is when we design these clinical trials and we have them ongoing, we get data from the lab, we actually can make changes, and you'll see whether it was by good fortune or whether for scientific principles, it worked. So to summarize, I think this would convince you that actually the vaccine was immunogenic, at least in primates, but you needed to do it by the route was critical, it was IV. The dose, which was the same dose he had used in sub-Q and ID, seemed to work well. And we had responses in T cells, in blood, in liver. I didn't show you the spleen data, but what you're getting is systemic activation. There are spleen-specific cells, and maybe the schedule's important. So let me make a little bit of a break here in terms of what standard vaccines do and what we did here with the IV. So when we give almost any vaccine, we give that is a sub-Q or IM injection locally in an arm, and in the case of BCG, it would be in the skin, is you get, if you track that vaccine, we do this all the time, we label vaccines and track where they're going, they all invariably track to the draining lymph node, and that's where you get priming. But they actually don't extend much beyond the draining lymph node unless you were giving something like yellow fever, a live virus that would go everywhere. So you're getting most times you're getting that one lymph node to do your priming. When you give it intravenously, you're accessing the whole immune system, right? You're getting multiple lymph nodes, but importantly, you're probably accessing the spleen, which is the largest lymphoid organ, and you're probably getting priming in the liver, which I think there was some heresy of whether you actually, that's an inductive organ for T cells. That also happens to be the organ by which you need cells for malaria. So you might extend, well, if you gave intravenous, you might get T cells into the lungs. If you had a TB vaccine, what do you need for a TB vaccine? You might need T cells present in the lung at the time of infection. So it tells you delivery is actually quite important. The issue, of course, is, there are no vaccines that are given intravenously, but from a scientific perspective, it makes sense that you get systemic priming. Now, if those of you who do therapeutic cancer vaccines, is there any reason why you would not want to potentially use an intravenous route rather than just a local route if you can access these other organs and get more of a robust response? So based on the primate data, we thought we had evidence to try intravenous immunization with this vaccine in humans. And so at the Vaccine Center, this was our first foray into malaria vaccines. And the primary objective, of course, is safety and tolerability given by intravenous immunization. The secondary objectives is, does it work? Is it effective? And can we measure immune responses that might tell us something about why it's working? And so this was the design. you started out, the FDA said, even though 135,000 had been given sub-Q and ID, and that's what we did in monkeys, they said if you're giving it intravenously, we want you to start lower. So we started with 2,000. The 2,000 is probably similar to what was in this piece of tape. But at any rate, we did 3 volunteers, and we gave 2 immunizations, and if it was safe, we went to 7,500. You know, I think this is good clinical practice. And we gave either 6 or 4 immunizations, then we went to 30,000. They got 6 or 4 immunizations at monthly intervals. And then 3 weeks after their final immunization, they're challenged, and we bring a group of subjects that had no vaccine as controls, and you challenge them. And then we had the group that the data was based on. It was 135,000. And these groups were originally supposed to get 6 immunizations, and this group would have gotten just 4 immunizations at monthly intervals. So this is where we made the change, where instead of giving 6, we gave 5, and we made that 8-week interval between the 4th and the 5th, based on the data we had from primates. So in the middle of the study, we just eliminated a vaccine and made that interval. And again, we had new controls, and we challenged them. So what you do is we go to Walter Reed. They have a beautiful insectary. And mosquitoes, 5 mosquitoes are placed on for 5 minutes. They're taken off. They're dissected. They look to see, did the mosquitoes take a blood meal, and do they have between 10 and at least 10 to 100 sporozoites in their salivary gland. And if they meet those criteria with all 5 mosquitoes, we feed them bagels, and they go home for a week. And then they come back because they come back, and in that phase, you're monitoring them every day by blood smear. In this phase, historically, what had been done is the Army always would do this and send them into hotels in Bethesda. So I always marveled at that you had your family on a vacation, and the Hampton Inn has half the hotel are having people that are infected with malaria, where people are every night. But we brought them into the NIH, and you can imagine, the NIH didn't know what to do. I mean, they said, you got people with malaria every night, and it was between, I think, fear and interest, you know, kind of watching a bad car accident. But at any rate, and all of us, I slept on call, Barney Graham slept on call. I mean, Barney is a very senior investigator. But we watched them every day. They would go to work, and then they would come back, and if they got malaria, we would treat them. We've subsequently we now do this by PCR. We don't even do blood smears. We bring them in the morning. They get a PCR done, they go to work. We don't admit them. This is all done as outpatient. The Army is still admitting people to the hotels and doing blood smear, but now you do it with PCR, and you catch them 2 days before the blood smear. And in one trial, the people who ended up getting malaria weren't even, really had no symptoms at all. So that's the model. And you're always wondering who signs up for this. So there is some remuneration for the inconvenience of doing this, okay? So here's the Kaplan-Meier curve, and you can see that these are all those who got low dose, 7,500 or 30,000, and were challenged 3 weeks later. And so this is the controls. All 6 unvaccinated subjects were infected, and of the 17 vaccinated subjects, only 1 of 17 did not get infected. So the low dose didn't work. When you went to the higher dose at 135,000, given 4 times at monthly intervals, 6 of 9 subjects did not get infected. In the 5-dose group, where we had that 8-week interval, none of the 6 subjects got infected. So 12 of 15 at 3 weeks did not get infected. 1 of the controls did not get infected. We brought that person back later, and they were infected because this became of enormous interest. Was the person taking chloroquine? Was there some cool, innate, at that point you get excited potentially they've got something that protects them, but ultimately, that person was infectable. But at any rate, I think you get the idea that this was statistically significant. So this was the proof of principle that this vaccine, given by the IV route, could lead to protection. So here's some immune responses. This is after the final immunization, 2 weeks later, the peak, 1 week prior to challenge. And you can see that for titers of CSP antibodies, that's that dominant protein, you get, through an increase in dose, that you get increase in these CSP titers. And you just don't have enough people to make any claims, but it appears that if you're protected, you had higher titers than these 3 people in this dose group that wasn't protected. So there's a dose effect for CSP titers, and these antibodies, there's a functional test to say not only do you have titers, but you can add the serum and show that it prevents invasion of the sporozoites into hepatocytes in vitro, so they're functional. So we have at least an antibody response that might predict short-term protection. When you look at the T cell responses, these are sporozoite-specific T cells after the final immunization, before challenge. You also see there's a dose response where at the highest doses you see CD4 cells that make any of these cytokines, gamma-2 or TNF, although you can see that one of the subjects that wasn't protected had just as high responses as those that were protected. So you just don't have enough people to make any claims other than the fact that, again, dose seems to predicate an increase in response. We looked at CD8 responses. So this was the whole deal. We thought this vaccine is mediated by CD8 cells in the liver. We found something interesting. We saw something interesting, is in the group in which all 6 people were protected, 4 of those people I couldn't detect CD8 responses in the blood. So what this meant was 2 things. One is in humans, CD8 responses are less important than the 19 Nature papers from mice that showed that that's how it was working, or we're looking in the wrong place. And I'm going to show you slides later that, in point of fact, and I still can't answer this question for you until I can get liver biopsies from people, but are the cells in the liver? But that was a wake-up call for either changing the way we think it works or we're looking in the wrong organ. So to summarize, it was well tolerated. It was very safe and well tolerated. There were no local infections. It actually probably has a better safety profile in this small number than almost any vaccine you could pull out. Even giving alum and hepatitis into the arm has more side effects than this vaccine. So it was a proof of principle, and that vaccine dose and interval were critical. So there's 3 things you take from this now. You want to try to understand why it's working, and more critical for any vaccine person, is it durable and would it mediate protection against other strains? So in terms of immune correlates, this is a picture of how we measure T cells. This is multi-parameter flow cytometry. This is a technique that's been worked out by my colleague Mario Roeder, in which you can do up to 18 different parameters on a single tube of cell to look at various cell surface markers, phenotype, and function. So here this just tells you we can look at CD4 cells, CD8 cells, NK cells, gamma delta cells, and mate cells. So both adaptive and innate cells. We can gate on them. We can look, are they memory? Are they central memory? Are they effector memory? And then do they secrete interferon, IL-2, TNF, and perforin, effector molecules? And then you can add receptors, chemokine receptors for homing, all these phenotypic and exhaustion markers, and then you can add a marker to see if they proliferate. So I'm not going to go into this into excruciating detail. I'm just saying with 18 colors, you're starting to get some real functional information. There's a new machine that we have now that will do up to 30 colors. So it's an efficient way to really understand as much as you can about that cell. And so when you use this tool, we take the total cells after an immunization, we re-stimulate them with sporozoites, and then we do the flow cytometry. And when we gate on CD4s and CD8s, and those are gamma delta cells, and these are mate cells, and these are NK cells, which are more innate type cells, you can see that the sporozoite stimulation, you get CD4s and CD8s, but you get this high frequency of gamma delta cells, a few mate cells, and some NK cells. So you're inducing multiple effector cells that all can secrete interferon gamma. And it raises the possibility, is there more to this than we thought? Is it not more to just CD8 cells in the liver that we thought? So the question is, are they actually sporozoite specific? And so to do that, what we do is we take the peripheral blood and we actually sort out each of these populations, CD4, CD8, gamma delta, mate, and NKs, and we add them back with their own antigen-presenting cells that we pulse with sporozoites and say, are they stimulated? Because that would tell you directly that individual cell is specific. And what we found something that was very interesting. So CD4 cells, when you add to an antigen-presenting cell that has the sporozoites, they make interferon gamma. They're specific. There's nothing else in that well. CD8 cells, the same thing. Gamma delta cells, the same thing. You add gamma delta cells, you add the APCs. But now you don't see any activity from mates and NKs. So when we were measuring in that whole culture that mates and NKs were making interferon gamma, they weren't directly specific. They were bystander cells, because if you added CD4 cells into these, they now make interferon gamma. And that occurs because the CD4 cells are making interferon gamma that induces IL-12 in that culture and causes those other cells which express IL-12 receptors to make interferon gamma. So what that tells you is you have antigen-specific cells, Then you have other cells, and these cells can all sit in the liver, and they all make interferon gamma. And the question is whether it's direct or it's indirect. If you have a cytokine that's going to kill something, might it be possible that there are multiple effector cells that could be mediating an effect? And if you go, there's a bunch of mouse literature to say that if you gave the sporozoite vaccine and deplete CD4 cells, you lose protection. Deplete CD8 cells, you lose protection. Deplete gamma delta cells, you lose protection. Or NK cells, you lose protection. So there's mouse data that suggests that they could all have a role. So this is probably more detail than you wanted to know, but I'm just saying it starts to open up some additional possibilities that potentially we haven't thought of, but we have the tools to kind of look at this. And I'm a T-cell biologist, you know. All right, but I must say at this point in my career, I'm far more interested in the Kaplan-Meier curves than I am that data. But you know, depending on which audience you're talking to, a lot of people would rather see that data. I think the other thing is we just did a kinetic with each immunization after the first, second, third, fourth, and fifth, and you can see something interesting. The peak in the blood is after the first immunization. And after each subsequent immunization in the blood, there's not an increase until we did that 8-week interval between the fourth and the fifth, and then you get a boost. So we have started to look at, we have to look after the first immunization to see what those peak responses are. But our guess is that most of the responses might be going to the liver. They don't come out of the liver. So there doesn't appear to be boosting within the blood. Same thing when you look at CD8 cells, the peak is after the first, and then in the blood there's no boosting until we have that final immunization where there's a bit of a boost. In terms of looking at those innate cells, I showed you that gamma delta cells can respond to the sporozoites. And so when we looked at just the frequency of gamma delta cells after each immunization, they do go up. So this is, I think eye-opening for us. And it turns out that gamma delta cells respond to this phospho antigen called HMBPP that's common to TB, listeria, and malaria. And so you start to think maybe gamma delta cells back in the day might have been to these type of infections a cell that could respond immediately to make interferon gamma. And over the years, we've gone to more adaptive immunity with CD4s and CD8 cells, but it starts to get interesting of what's the role of gamma delta cells to this infection. And so gamma delta cells expand, and there's many clonotypes of gamma delta cells, so it turns out that the one that expands is this V delta 2 V gamma 9. And so knowing nothing about gamma delta cells, we now are spending a fair amount of time trying to understand their role. Are they a marker of immunogenicity? Do they have an effector role? Or do they have some role in the initial priming? And I don't know the answer to any of those, but we're spending a lot of time probing that. So to conclude, I showed you the kinetics, the T cells response, the responses peak after the first immunization. That 8-week interval might be important at the end. You get multiple populations of CD4, CD8s, and gamma deltas, and the gamma delta cells expand. So now, the final thing I'll close with is durability. So what we did is we brought, this was the subjects who I showed you were protected when they were challenged 3 weeks later. We brought back 3 of the protected subjects in this group and 3 of the protected subjects in this group, and re-challenged them 5 months later, and 2 of the 6 remained protected. So this would tell us that, okay, there is some durability, but we're not there yet. That dose was not sufficient for what we would consider to be high-level protection. This just guided us to that we needed to go higher. So 4 or 5 doses at 135,000 was not sufficient. So we designed a new trial. And so we said we would just double the dose. Now, we doubled the dose because we said we were doing pretty well, and since we were trying to be careful, we doubled the dose. I can tell you now we're up to 900,000, and now their new studies plan to go to 1.8 million. And so we were trying to be very careful at doubling the dose. If you think about how viral vaccines are given, you wouldn't be doubling viral vaccines. You'd be going up by logs. But I think there was concern, obviously, for safety, and so we were kind of going up slowly. But I think I should tell you we're at least 10 to 20-fold higher than this right now. But we did 270,000 times 4 doses, and we actually challenged them, either a group 3 weeks later and brought whoever was protected back 6 months later, or we did the clean memory experiment. because it's possible that this challenge might have influenced immunity. So we just said, what happens if we just wait 6 months? The second thing we did is we had a group where we just repeated what we had done in the first study to make sure is a positive control. We gave 135,000 5 times since we had high-level protection there. We tried to simplify the regimen, so by doubling the dose, we tried to go down from 4 to 3 doses. And then we did one more thing, was IV essential, or was it just more efficient? So we knew ID and sub-Q didn't work, so we said, let's try it by IM, but let's give roughly 10 times the dose. IM on the same 4-dose regimen. Because if IM could work, it might be easier to administer, especially in newborns or infants. And so I'll just show you, I think, the data with the 270,000 given 4 times. And so when this group was challenged, or originally 12 subjects, but we only kept 9 in the study, 7 of 9 were protected at 3 weeks. So we were thrilled with that, and one of the controls, again, didn't get infected, but this is roughly 80% protection. So by doubling the dose, we had high-level short-term protection. When we brought this group and challenged them 6 months later, we had 6 of 11, or 55% protection. When we brought 4 of these people back, 3 of 4 remained protected. So we now have protection of 55% at 6 months. We brought back 5 of these people a year later and challenged them, and all 5 are protected. So overall, with a 4-dose regimen at 270,000, we're at 55%. So we're getting there, but as I've shown you, dose is critical for this vaccine. And so now we're increasing the dose to try to, the goal is to get 80 to 90%, at least 6-month protection. And then, obviously, the question is how this would work in the field. Just to show you over the course of a year what the immune responses do in this 4-dose group, and you say this is the CSP titers. So you see after the final immunization they peak, and then over the course of a year they go down almost a log. So antibodies are quite low at that 1 year when people are protected. By contrast, when you look at these gamma delta cells, they go up and they remain stable through the whole course of the study. CD4 cells peak, and then they also go down over a long period of time, and CD8 cells are very low to begin with and also go down. So at the end of a year, you have very low antibodies and low T cells in the blood, and you have gamma delta cells in the blood. And so the summary is that we are getting there. We have 6-month durable protection, about 55%. I didn't show you the correlates data, but what correlates with protection in this study at 3 weeks or at 6 months was the titer of CSPs or the frequency of gamma delta cells. And so then the question is, how is it really working? If antibodies are really low at 1 year, what could it be? And so I'll go back to the monkeys. So this was just another monkey study. We had 4 animals. We gave them IV immunization. And we looked 2 weeks after their final immunization. You can see that 2 of the monkeys had almost 2% in the blood, and 2 had very low responses for interferon gamma. When we looked at these monkeys 4 months later, you can see they contracted 10-fold, and these still remained low. But when we looked in the liver, so we looked in the blood and the liver at the same time, the animals were euthanized, you can see that these 2 who had low responses in the blood all along had robust responses in the liver. And this one, where you can detect it in the blood, also had very high responses in the liver. So this tells me, I think, that the blood might not necessarily tell you what is happening in the liver. And if you then look in the liver and look at a pie chart of all these different populations I've been telling you, this is the pie chart of the different populations of CD4s, CD8s, NK cells, gamma delta cells. You can see in the liver that the CD8s are 50%, the CD4s are less, and these are NKs and gamma delta cells. And when you look after a vaccination, you can see that we get huge numbers of CD8s and NK cells and gamma delta cells. You have all of these populations in the liver. So is it conceivable that the cells are primed in the liver and they stay there and you have multiple populations? And we're never going to know that from humans. We just can't get liver tissue. But we can use the primates to try to understand mechanisms. And so this just tells you maybe a cartoon of what I think might be going on. The sporozoites get into the sinusoids, they transverse a Kupffer cell, and they find their way into a hepatocyte and they expand dramatically, and then you have all of these cells that get, the CD4 cells get activated by Kupffer cells. They might make IL-12. You have NK cells and gamma delta cells that can respond to IL-12. You have CD8 cells that can potentially recognize the hepatocyte directly. You might have multiple effectors that could secrete interferon gamma to mediate what I consider a cumulative effect. So finally, what about heterologous protection? I didn't do this work. This is a study done in Mali. And so what was done is they were given 270,005 doses. This was done by Patrick Duffy during the non-rainy season. And then he, over the course of the rain, after the vaccination was completed, the rain started. And he showed that this 5-dose regimen gave about 28% protection in the field at 6 months. What you'll notice is the antibody response to 5 doses is much lower than what 5 doses of the same vaccine would do given it Walter Reed. So you know that in the field that people that have been exposed with malaria make much less antibody than they would in naive subjects. So that's a daunting prospect, because if we're taking this vaccine and we're going to Africa, they might have preformed antibodies that could bind the vaccine. They might have regulatory cells from prior malaria exposure that would alter the priming. So there's really only one trick that you can do, is you can just increase the dose. But it tells you that going into Africa, into people that are already exposed, or at least adults, might be different than a naive population in the United States. So how do we get around that? When I saw that data, I said, what we need to do is we need to go into a naive population. And so that population would be infants. So when maternal antibody passes at 5 months, we would pick infants between 5 and 12 months of age, where presumably they're going to have less exposure. We take the vaccine and we give it to that age group for 2 reasons. One, they've had less exposure and they could respond better. And 2, they're the ones that are at risk of dying. So the protocol has been written. We hope to start that in Western Kenya, do a dose and age de-escalation in March, and then the efficacy trial would be started in the fall. So the lessons, getting back to how you'd apply this, and maybe this applies, is when you have something complicated with multiple antigens like malaria, does it apply to other things? So when I look at things requiring T cells, it's always magnitude, quality, and breadth. You need numbers. You need cells that can secrete effector functions that aren't exhausted. I've shown you that would you take advantage of the breadth of effectors? Might you engage other populations like gamma delta cells and NK cells? With TB and malaria, you have multiple genes. Often we go with one target. We're using one antigen or 2 antigens. Is that the right thing to do? Location is critical. For malaria, you need the cells in the liver. TB, you need them in the lung. If you want it for HIV, there's data that suggests that if you give vaccines intravenously, you can get a very high frequency in the vaginal mucosa in monkeys that might mediate protection. So route is critical. What we've done now, based on these data, is we have monkey studies now where we give BCG by aerosol or intravenously to try to get cells, rather than the liver, we want them in the lung. I told you that I think preclinical models like non-human primates are critical for kind of predicting what's going on. And the human challenge models are critical for rapid development. And there's a movement afoot to try to develop human TB challenge models, because you can really test your vaccine and get data on relatively small numbers of people very quickly. So let me just close. These are monumental group efforts. So first of all, you have to thank the volunteers who are willing to take a sporozoite vaccine intravenously. The first study, you know, especially that first study, now all our studies are filled with graduate students, many of whom who do malaria research, because I think they read the literature and think they're going to get protected and are happy to have the remuneration. So then you start with the Steve Hoffman and Kim Lee Sim and their colleagues at Scenarius. Steve was the visionary who decided to make the vaccine and take the time and effort and cost to perfect it. The Walter Reed Insectary, in terms of doing all our challenges with Silas, Dan Somolas, and Lindsay Garver. And at the Vaccine Center, the memory studies I showed you were really led by Kirsten Leik and Andrea Berry at the University of Maryland. Julie Ledger and Barney Graham run our clinic, and they were heavily involved in the first study. Mario helps me with all the flow. Martha does the statistics. In my own lab, Andrew Ishizuka, a wonderfully talented graduate student does the flow cytometry. Ozza, Neville, and Kalen also helped in the analysis of the malaria-specific responses, and then the clinical center provided the core lab. So with that, I'll take questions.
Bob, that was terrific. And while they're getting microphones out to people, please raise your hands if you're interested in questions. I wanted to ask you, so giving IV vaccines is a new challenge for us. And you mentioned tumor immunology, but you could imagine we could make it practical in other centers. You're also really involved in adjuvant production. And adjuvants, as you know, go in with vaccines, and they're sort of old school adjuvant is to put stuff locally and keep it together, not necessarily cross-link it. But when you give it in the blood, it gets separated. So how are you going to address that?
Yeah, so that's a wonderful question. So you don't need IV for everything. For the vaccines where you might need low-level antibody responses, which most of our vaccines work through, you wouldn't need to do this. The question is the big three, malaria, HIV, TB, they all represent challenges or tumors in really getting much higher levels of antibodies or mutated antibodies or high levels of T cells. There you need to be a bit more clever. So what Wes is asking is if you were going to do an adjuvant delivery, you would have to have your adjuvant linked to your protein. So you'd either give, like in the case of an ad vector, you have your antigen expressed within the ad vector, and the ad vector induces innate responses. So that's packaged together. If you're going to use peptides or proteins, what we do is we physically link toll ligands so they're delivered together, because what you're right is that if you didn't link them, you could get antigen presented without any innate stimulation, and you would get tolerance. And then the adjuvant effect could be synchronous or could not be synchronous. So when we do intravenous, at least with those deliveries, they must be linked. And I'm not suggesting you do that for things other than potentially therapeutic cancer vaccines where you might need large numbers of T cells and where the risk profile changes, where you're willing to use adjuvants that might be more inflammatory because you need to get cross-presentation. So you can't do IV probably protein and adjuvant yet in 5-month-olds in Africa, but you certainly might be able to do it therapeutically in cancer vaccines. Yeah, so the question is can you use hepatitis B as your delivery? So it's a good question. The issue is the time and care it takes for them to isolate the sporozoite. I think the only thing you potentially could do is perhaps adjuvant it if you added something. So suppose gamma delta cells or NK cells were important. Could you give something like alpha-gal ser? Could you give something to just add to it? I would worry that any manipulation of the sporozoites to bind to something else might impair its integrity. I mean, frankly, there's still a group of disbelievers out there, despite all of this data, that what's in the vial is crap, or it's a little better than crap. I mean, and so I think those are people who dealt with these for a long period of time. So I think they're saying these are very delicate. So I think it would be hard to do that type of manipulation. Where I think this goes is this is a proof of principle, and I think, like I said, someone like Stefan Kaplan really has worked on this for years. Is can you do a bit better by having genetically attenuated sporozoites, which actually are live and go through the liver? It might give you a more uniform and a deeper breadth of responses, and then they turn off. And so I think these type data set him up well for those types of studies, and I think those are ongoing now.
This may be thanks for an excellent talk. First, this may be complementary to the last question. I'm trying to think of alternate delivery routes, and thinking of places like the Cambodian-Thai border or rural Kenya, and trying to think, how would you possibly get 5 intravenous doses into a kid under age 2 or 3 when they struggle to get 1 or 2 DPTs IM?
Okay, so you've asked so normally Steve Hoffman takes much more of this than me. I try to be scientific about this. But yeah, no, the bullets are what's in the vial. This is crazy. How are you going to do that? Okay, so in Kenya, I think Steve has gone into Mali. Patrick Duffy did the study in Mali. They did 490 consecutive intravenous immunizations in adults without a problem. I think in adults, it's not a problem. Our study served three functions. This infant study is one, the scientific issue of them being naive. Two is try to prevent them from dying. But three is, can you do it? So I was just in Kenya 2 weeks ago in Siaya. I have some travel pictures, and I'll show you what the infrastructure is in Siaya. And that's the question you really want to know is feasibility. And so can we, in 400 infants, can we do an intravenous immunization? That's why I pushed Steve to do the IM. He didn't actually want to do it because I was using 2 million per dose, and I was killing him because he had to make a lot of sporozoites. So that told us that there was something unique, that the IV was really required, okay? One issue is, could you use microneedles eventually? Could you get a delivery technology where you would have the sporozoites and you'd get microneedles to deliver them through the skin into the blood, right? But I think the study in Kenya will tell us the feasibility. Can we get an infant not to squirm, and can we get the vaccine delivered? And, if we can't, that'll tell you, right? So that's why we're doing the study. I can tell you that I've made this clear to my colleagues at the CDC who have set up the site in Siaya, that the most critical people by far in this study are going to be the people that are going to be administering the vaccine, right? That's really the whole key to doing it. But the important thing is proof of principle. If I came here and said I was going to do intravenous from the monkey study, you all would have looked at me like I was crazy to begin with. Okay, so there's been 10 studies since then, so at least we've learned things. We can get protection, we can map antigens, we can look at correlates. So let's just, as long as it's safe, let's do the proof of principle.
Bob, thanks for a great talk. When should this stuff about route and intravenous immunization is fascinating. For what pathogens, thinking more broadly than malaria, should we be thinking about combinations of intravenous and local? So, for example, in STIs, including HIV, one can think about many of these that, where, you want to give mucosal protection, but then they sit in the, the ganglion, like herpes. So should we be thinking about that, and when and why?
Yes, so Dave Mazbut, this is his experiment. He gives listeria, recombinant listeria. 8 weeks later, he gives recombinant VZV, and 8 weeks later, he gives recombinant Ad5 in sequence to SIV gag, and he gives them intravenously in monkeys, and he shows that he has, I think, between 5 and 10% CD8 cells in the vaginal mucosa in monkeys. And when he did his first experiment, like half of the animals to an SIV challenge were completely protected. And so the idea is that he had a sufficient frequency at the site, or at the beachhead. And so you might imagine for HSV is, how do you get cells, right, into that area? So you will get, from a viral vector, you probably will get some cells to migrate there, but you need to either pull them in by some sort of boost. So maybe you give a systemic immunization, and then you have to actually go locally where there's antigen. Because where do the T cells go? The T cells go where there's antigen. So can you do that? Or would you give it intravenously? And then it just becomes a safety issue, right? It turns out the sporozoites probably induce not only Th1 responses, might induce Th2 responses. So we've not had a safety issue with irradiated sporozoites, but if you give, Ad5 intravenously, then it might be different. But the idea, is location matters and route matters. So for TB, I think you would agree, doesn't it make sense that we would give our TB vaccines by aerosol to generate cells in the lung? So when you get infected with TB, there's no weight of migration of cells into the lung. The cells are there. And so whether BCG-IV is a proof of principle could work, we'll test. But certainly it changes your idea of delivery. It's nothing profound, I don't think. It's just doing it and showing it's safe.
Well, we're out of town. If you have more questions, please come up front. But